Okay. Chapter 11 is about Mendel. In the gene idea, Mendel discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. He chose to work with peas because they had many varieties with distinct features or characters, such as flower color, seed shape, seed color, and so on. These characters or character variants are called traits. He could also control the mating between plants, so that made it very easy to do his experiments. Okay, so he's crossing pea plants, so he's taking a purple plant and he's crossing it with a white plant here. Mendel chose to track only characters that occurred in two distinct alternative forms. We know that these forms are now called alleles. He also used varieties that were true breeding, plants that produce offspring of the same variety when they self-pollinate. So true breeding, these are going to be either homozygous dominant or homozygous recessive individuals, not heterozygous. In a typical experiment, Mendel mated two contrasting true breeding varieties, a process called hybridization. Like, for example, he crossed purple flowers with white flowers. The true breeding parents are the P generation or the parental generation, and then the offspring of the parental generation are called the F1 generation. And then if you take two F1 individuals and you self-pollinate or cross-pollinate, then you end up with the F2 generation. When Mendel crossed contrasting true breeding white and purple flowered pea plants, all of the F1 hybrids were purple. When Mendel crossed the F1 hybrids, many of the F2 plants had purple flowers, but some had white. Mendel discovered a ratio of about 3-to-1 purple to white flowers in the F2 generation. Okay, so these are purple true breeding flowers, so they are homozygous dominant. These are your white flowers, those are homozygous recessive. They are not heterozygous. You know that because when you breed them with themselves, you self-pollinate. The purple always gave purple flowers and the white always gave white flowers. Okay, so you cross the purple with the white. The F1 generation had all purple flowers. These are known as hybrids. These are heterozygous purple flowers. If you take two heterozygous purple flowers and you cross them, you end up with the F2 generation, where he saw the very classic 3-to-1 ratio of purple to white flowers. Mendel reasoned that in the F1 plants, the heritable factor for white flowers was hidden or masked in the presence of the purple flower factor. He called the purple flower color a dominant trait and the white flower color a recessive trait. The factor for white flowers was not diluted or destroyed because it reappeared in the F2 generation. Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits. What Mendel called a heritable factor, we now call a gene. So here are some of the traits that he looked at. So flower color, seed shape, pod color, flower position, and stem length. All of which only had 2 alleles for each character or each trait. Mendel developed a model to explain the 3-to-1 inheritance pattern he observed in the F2 offspring. 4 related concepts make up this model. First, alternative versions of genes account for variations in inherited characters. We know that these alternative versions of genes are called alleles. For example, the gene for flower color in pea plants exists in 2 versions, one for purple flowers and the other for white flowers. Each gene resides at a specific locus or location on a specific chromosome. So these are a pair of homologous chromosomes, and you can see where the allele for flower color is. On this homologous chromosome, the allele happens to be purple, and on this one, the allele happens to be white. Second, for each character, an organism inherits 2 alleles, one from each parent. Mendel made this deduction without knowing about the existence of chromosomes. 2 alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation. Alternatively, the 2 alleles at a locus may differ, as in the F1 hybrids. Third, if the 2 alleles at a locus differ, then one determines the organism's appearance, which would be the dominant allele, and the other, the recessive allele, has no noticeable effect on the appearance. So the dominant allele masks the recessive allele, and it is the dominant phenotype or appearance that shows up in the individual. In the flower color example, the F1 plants had purple flowers because the allele for that trait is dominant. Purple was dominant to white. Fourth, now known as the law of segregation, the two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. Okay, so this has to do with meiosis, where the homologous pairs separate during meiosis 1, and then the sister chromatids get ripped apart during meiosis 2. Thus, an egg or a sperm gets only one of the two alleles that are present in the organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. Okay, so here is a Punnett square looking at the offspring of the F1 generation. So the F1s are heterozygous. So we're crossing two heterozygotes. And remember, Punnett squares give you the predicted results of the offspring. So this is a prediction. If you cross two heterozygous plants, you would expect to see a 3-to-1 ratio of purple to white in the offspring. Mendel's segregation model accounts for the 3-to-1 ratio he observed in the F2 generation of his numerous crosses. The possible combinations of egg and sperm can be shown using a Punnett square, a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. A capital letter represents a dominant allele and a lowercase letter represents a recessive allele. For example, capital P is the purple flower color allele and lowercase p is the white flower allele. An organism with two identical alleles for a character is said to be homozygous for the gene controlling that character. So you could be homozygous dominant or homozygous recessive. If an organism has two different alleles for a gene, it's said to be heterozygous for the gene controlling that character. Unlike homozygotes, heterozygotes are not true breeding. Okay, so homozygous is the same as true breeding, heterozygotes are the same as hybrids. Because of the effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. Therefore, we distinguish between an organism's phenotype, its physical appearance, and its genotype, or genetic makeup. In the example of flower color in pea plants, capital P, and capital P lowercase p plants have the same phenotype, but different genotypes. Okay, so you can see here that here are 3 purple, phenotypic ratio, 3 purple to 1 white. But if we look at the genotype, we have 1 homozygous, 2 heterozygotes, and then we have our homozygous recessive white plant, white flower. So just by looking at the phenotype, we can't determine whether these individuals are homozygous dominant or heterozygous. In cases like this, we would want to run a test cross. What a test cross does is it breeds your individual, your phenotypically dominant individual with a homozygous recessive individual. So this is a test cross. So we have a purple flower, and we don't know whether it is homozygous dominant or heterozygous. So what we can do is we can take that flower and we can cross it with a homozygous recessive individual, the white flower in this example. Now, if once we cross it, we end up with white flowers in our offspring, that means that this purple flower had to be heterozygous in order for the white flower phenotype to show up in the offspring. If we get all purple, then our flower is most likely homozygous dominant. Mendel derived the law of segregation by following a single character. The F1 offspring produced in this cross were monohybrids, individuals that are heterozygous for one character. A cross between such heterozygotes is called a monohybrid cross. Mono means one, it's looking at one trait or character. Mendel identified his second law of inheritance by following 2 characters at the same time. Crossing 2 true breeding parents differing in 2 characters produces dihybrids in the F1 generation, heterozygous for both characters. A dihybrid cross, a cross between F1 dihybrids, can determine whether 2 characters are transmitted to offspring as a package or independently. So this this ties to gene linkage. So if we were to do a dihybrid cross, we would need 16 squares, okay? And in this case, we're crossing two heterozygous individuals, and when we cross two heterozygous individuals, remember the predicted ratio is going to be 9 to 3 to 3 to 1. Now, if we get something very different from that, let's say that two of the phenotypes show up way more than two of the other phenotypes, then that probably is going to lead us to believe that there is gene linkage. Genes are inherited together because they are close together on the same chromosome. The results of Mendel's dihybrid experiments are the basis for the law of independent assortment. It states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. This law applies to genes on different non-homologous chromosomes or those far apart on the same chromosome. Because remember, those far apart on the same chromosome have a high frequency of crossing over. So it's as if they act like they don't reside on the same chromosome. Genes located near each other on the same chromosome tend to be inherited together. We call those linked genes. Mendel's law of segregation and independent assortment reflect the rules of probability. When tossing a coin, the outcome of one toss has no impact on the outcome of the next tossed. In the same way, the alleles of one gene segregate into gametes independently of another gene's alleles. The multiplication rule states that the probability that two or more independent events will occur together is the product of their individual probabilities. This can be applied to an F1 monohybrid cross. Segregation in the heterozygous plant is like flipping a coin. Each gamete has a half chance of carrying the dominant allele and a half chance of carrying the recessive allele. Okay, so that's represented here. Flip a coin. There's a 50% chance that the sperm will get the dominant allele. There's a 50% chance that the sperm will get the recessive allele. Same thing goes for the egg. So, chance of those occurring together, 1/2 times 1/2 means we would get 25% chance to get big R, big R. 1/2 times 1/2, same thing, 25% chance of getting big R, little r. Same thing, 25% chance getting little r, big R, or little r, little r. I'm going to skip the addition rule. We can apply the rules of probability to predict the outcome of crosses involving multiple characters. A dihybrid or other multi-character cross is equivalent to two or more independent monohybrid crosses occurring simultaneously. In calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. For example, if we cross F1 heterozygotes of genotype big Y, big R, we can calculate the probability of different genotypes among the F2 generation. Okay, so we want to know the probability of getting big Y, big R, big R. So remember that we are crossing two heterozygous individuals. So if we do a Punnett square of big Y, and cross that by big Y, little y, we have a 1/4 probability of getting big Y, big Y. Likewise, we crossed big R and a little r, so the probability of getting big R is also 1/4. So the probability of those two events happening simultaneously is the multiplication rule. 1/4 times 1/4 gives us 1/16. So we have 1/16 chance of getting big Y, big R, big R. If we want to know the probability of getting big Y, big R, little r, we just need to change the first part. So the probability of getting big Y, little y is 1/2. In our Punnett square, we could have gotten a big Y, little y, or a little y, big Y, so that's 1/2. Probability of getting big R is still 1/4. Probability of two events occurring simultaneously, we multiply, so we get 1/8. We can do another example. Looking at this cross, we can calculate the probability of offspring showing at least two recessive traits. So at least two recessive traits. These are all of the combinations that give me at least 2 recessive traits. Here it's P and Y, here it's P and R, here it's Y and R, here it's all 3. Okay? So, probability of little p is 1/4, because big P by big P gives me 1 out of 4 boxes with little p. Probability of little y is 1/2, because we are crossing big Y with 2 little y's. So half my boxes are going to be big Y, and half my boxes are going to be little y. And then lastly, big R. We crossed a big R with a big R, so we get a half chance of getting big R. Probability of all those things happening simultaneously, multiplication rule, we multiply them together, we get 1/16. We can do the same thing for the rest of our genotypes, and then chance of at least 2 recessive traits, we would add those all up, and we would get 6/16, or 3/8. Not all heritable characters are determined as simply as the traits Mendel studied. However, the basic principles of segregation and independent assortment apply even to more complex patterns of inheritance. Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations: when alleles are not completely dominant or recessive, such as an incomplete dominance or codominance; when a gene has more than 2 alleles, multiple alleles; when a single gene influences multiple phenotypes. Okay, so let's take a look at each one of those. Complete dominance, that's what we've already been talking about. Complete dominance occurs when phenotypes of the heterozygote and dominant homozygote are identical. So your dominant allele completely masks your recessive allele. In incomplete dominance, however, the phenotype of the F1 hybrids is somewhere between the phenotypes of the 2 parental varieties. In codominance, 2 dominant alleles affect the phenotype in separate, distinguishable ways. Okay, so this is an example of incomplete dominance with snapdragons. So you take a red snapdragon and you cross it with a white one, you end up with pink flowers, pink snapdragons in your F1 generation. The pink phenotype is an intermediate phenotype between red and white. That's incomplete dominance. Codominance, I think, is a little bit later on, but codominance is different in that both alleles would show up. So if this were codominance and you crossed a red and a white flower, you'd get flowers that have both red and white spots or stripes, for example. Example of codominance, blood type, where A and B are codominant, or roan horses, where you have the red and the white horses that mate, and you get horses that have both red and white hairs. The relationship between dominance and phenotype. Alleles are simply variations in a gene's nucleotide sequence. When a dominant allele coexists with a recessive allele in a heterozygote, they do not actually interact at all. For example, dominant recessive relationships of alleles depend on the level at which we examine the phenotype. Tay-Sachs disease is fatal. It has to do with a non-functional, dysfunctional enzyme that causes the accumulation of lipids in the brain. At the organismal level, the allele is recessive. At the biochemical level, the phenotype, for example, the enzyme activity, is incompletely dominant. At the molecular level, the alleles are codominant. So if an individual is heterozygous, for example, their enzyme activity would not be as great as someone who is homozygous dominant. Dominant alleles are not necessarily more common in populations than recessive alleles. For example, 1 baby out of 400 in the US is born with extra feet or toes, which is a dominant trait called polydactyly. Most genes exist in populations in more than 2 allelic forms. For example, the 4 phenotypes of the ABO blood group in humans are determined by 3 alleles of the gene IA, IB, and little I. The enzyme adds specific carbohydrates to the surface of blood cells. The enzyme encoded by IA adds the A carbohydrate, and the enzyme encoded by IB adds the IB carbohydrate. The enzyme encoded by the little I allele adds neither. Okay, so here are my blood types. So this is blood type A. So the A carbohydrates are represented by triangles here. The B carbohydrates are represented by circles. If you have AB blood type, you have both carbohydrates on the surface of your red blood cell. If you're a blood type O, you don't have any. So remember, AB is the universal receiver, O is the universal donor. Because remember, these carbohydrates, they look foreign to people that don't have those carbohydrates on the surface of their red blood cells. Most genes have multiple phenotypic effects, a property called pleiotropy. For example, pleiotropic alleles are responsible for multiple symptoms of certain hereditary diseases such as cystic fibrosis and sickle cell disease. Some traits may be determined by 2 or more genes. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, Labrador retrievers. So Labrador retrievers, you can have black labs, chocolate labs, or yellow labs. So black is dominant to yellow. Black is dominant to brown, and yellow occurs because there's a second gene that controls the depositing of the pigment in the fur coat, okay? So in order to be black, a black lab, you would have to have at least one dominant allele for coat color and one dominant allele for the depositing of that coat color. So black labs would have to have at least one capital B and at least one capital E. Now, in order to be a chocolate lab, they would have to be homozygous recessive for coat color, but they would need to have at least one capital E for the depositing of that coat color. Versus my yellow labs, the yellow labs, it doesn't matter what the genes for the coat color are, because my yellow labs are homozygous recessive for the depositing of that coat color. So in this case, yellow represents an absence of color. Quantitative characters are those that vary in the population along a continuum. Quantitative variation usually indicates polygenic inheritance, an additive effect of 2 or more genes on a single phenotype. Skin color in humans is an example of polygenic inheritance. Okay, so here we have 3 genes which control skin color. So as a result, you can see all of the possible combinations that you could possibly get for skin color. So polygenic inheritance, more than one gene controlling a trait. Another departure from Mendelian genetics arises when the phenotype for a character depends on environment as well as genotype. The norm of reaction is the phenotypic range of a genotype influenced by the environment. The phenotypic range is generally broadest for polygenic characters. Such characters are called multifactorial because genetic and environmental factors collectively influence the phenotype. An organism's phenotype includes its physical appearance, internal anatomy, physiology, and behavior. An organism's phenotype reflects its overall genotype and unique environmental history. Okay, many human traits follow Mendelian patterns of inheritance. Humans are not good subjects for genetic research. Generation time is too long. Parents produce relatively few offspring, and breeding experiments, like the ones that Mendel did with his pea plants, are obviously unacceptable. And they would take too long anyways, even if they were acceptable. So basic Mendelian genetics endures as the foundation of human genetics. A pedigree is a family tree that describes the interrelationships of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Pedigrees can also be used to make predictions about future offspring. We can use the multiplication and addition rules to predict the probability of specific phenotypes. So here we have 2 separate pedigrees. On the left, we have widow's peak, which is this little peak in your hair on your forehead, versus no widow's peak. And it says, is widow's peak a dominant or recessive trait? Well, obviously the letters are already written in here, so obviously widow's peak is a dominant trait because capital W, lowercase double is shaded in. Let's see, capital W, or capital W, lowercase w shaded in. So all the ones with at least one capital letter are shaded in. But if we did not have any letters, the way that we would know that this is dominant is that every affected individual has an affected parent. Okay, so Widow's Peak, individual, parents both have it. Widow's Peak, individual, parent has it. Widow's Peak, individual, parent has it. Widow's Peak, parent has it. Okay, versus a recessive trait. So in order to know from a pedigree that a trait is recessive, you could have an individual that has the trait with two unaffected parents, like this one here, and these two here. Okay, so we know that attached earlobe is recessive. It's a recessive trait. Many genetic disorders are inherited in a recessive manner. These range from relatively mild to life-threatening. Recessively inherited disorders show up only in individuals homozygous for the allele. Carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal. Most people who have recessive disorders are born to parents who are carriers of the disorder. Like albinism. Albinism is a recessive trait. So you would have to have two parents that are heterozygous mating to produce a child that is albino, which again is an absence of pigment. If a recessive allele that causes a disease is rare, then the chance of two carriers meeting and mating is low. Consanguineous, there we go, between matings between close relatives increase the chance of mating between two carriers of the same rare allele. Most societies, as a result, and cultures have laws or taboos against marriages between close relatives because these rare genetic disorders can show up more frequently. Cystic fibrosis is the most common lethal genetic disease in the U.S., striking 1 out of every 2,500 people of European descent. The cystic fibrosis allele results in defective or absent chloride transport channels in plasma membranes, leading to a buildup of chloride ions outside the cell. Symptoms include mucus buildup in some internal organs and abnormal absorption of nutrients in the small intestine. Sickle cell disease affects 1 out of 400 African Americans. The disease is caused by the substitution of a single amino acid in the hemoglobin protein in red blood cells. In homozygous individuals, all hemoglobin is abnormal or sickle-shaped. Symptoms include physical weakness, pain, organ damage, and even paralysis. However, there is something called heterozygote advantage. So heterozygotes are usually healthy, but may suffer some symptoms. About 1 out of 10 African Americans has sickle cell trait, an unusually high frequency of an allele with detrimental effects in homozygotes. Heterozygotes are less susceptible to the malaria parasite, so there is an advantage to being heterozygous. So this is an example of why the homozygous allele is not going to disappear from the population. It's actually going to stick around with quite frequency where in places where there is a high frequency of malaria, because these heterozygotes make them less susceptible to malaria. Some human disorders are caused by dominant alleles. Dominant alleles that cause a lethal disease are rare and arise by mutation. Achondroplasia is a form of dwarfism caused by a rare dominant allele. So here is achondroplasia, which is a dominant trait. The timing of onset of a disease significantly affects its inheritance. For example, Huntington's disease, if you have Huntington's disease, which is a degenerative disease of the nervous system, you don't see any phenotypic effects until you are from 35 to 45 years old. And at that point in time, most people, if they're going to have children, have already had children. So they unknowingly pass on this recessive allele to their offspring. Once the deterioration of the nervous system begins, the condition is irreversible and fatal. Many diseases such as heart rate, diabetes, alcoholism, mental illness, and cancer have both genetic and environmental components. Lifestyle has a tremendous effect on phenotype for cardiovascular health and other multifactorial characters. So things like, how much you exercise and, what you eat can all have an effect on your phenotype. Okay. Genetic counselors can provide information to prospective parents concerned about a family history for a specific disease. Each child represents an independent event in the sense that its genotype is unaffected by the genotypes of their older offsprings. And I just realized that I misspoke right here. Huntington's disease is a dominant trait, dominant allele, not a recessive one. Sorry about that. Okay, that's going to conclude this chapter.